So bacteria has a special defense mechanism. So what happened here, once it is infected, okay, there is a specific region, okay, that will be incorporated, specific region of DNA of the virus, okay, will be incorporated. Incorporation, okay, I hope that you can see this. If not, then please open your own because this is PDF. Okay, I cannot like enlarge more. Incorporation of viral DNA into the bacterial DNA. So the bacteria has the capacity to copy, well, I use a simple word, to copy a specific region, which is the DNA of the virus into its DNA. Okay, you can see that it copies. Yeah. So spacer is actually the gene sequence of the virus that has infected the bacteria. Okay, I repeat this. So spacer is the gene sequence, specific gene sequence that has infected the bacteria. In this case, as an example, Streptococcus bacteria. So why does the bacteria do this? Because in future, if the same virus infected the bacteria, okay, it will recognize. So it serves as memory. Okay, so this gene sequence, which is called spacer, in the series of genes sequence of the bacteria, okay, it serves as a memory. It will memorize, okay, that it is the same virus that has infected the bacteria. So what happened if there is the same virus and it is detected by the bacteria, okay, there is what we call as Cas9 enzyme. You heard in the talk, we have Cas9 enzyme, Cas12 enzyme, Cas13 enzyme. So what is the function of this Cas enzyme? It can cleave, okay? So Cas9 complexes with the tracer RNA, okay? And CRISPR RNA duplex, okay? This is something that you have to read some more, okay? And then what is tracer RNA and CRISPR RNA? So the whole thing, the Cas9 and the tracer RNA and the CRISPR RNA, if this system finds the sequence of the spacer, okay, matching the tracer RNA, CRISPR RNA, it will cleave, means that if they find the virus, which the bacteria has the memory, so these complexes, okay, the Cas and the tracer RNA will cleave the virus. So sort of it will kill the virus. So that's how the defense mechanism of the virus, of the bacteria, sorry. Okay, so they are able to incorporate a series of genes into their own gene sequence. And in future, if there is a virus, the same virus which has which has specific gene that they have memorized, so they have these Cas complexes that can attack the virus by cutting. By cutting the virus means that the virus will be killed. Okay, so that is basically called the defense mechanism of the bacteria, the basis of what is the basis of CRISPR. So this is what happened. This is the Cas complex. Yeah? This is a Cas complex. Cas means the enzyme that cleaves together with the tracer and CRISPR RNA. So it goes like scouting inside the bacteria, and if it see the target, which is the virus, the same virus, so it can, this is the mechanism that it does, it cleaves, cut, yeah. So they cut from here and here. So once it is cut, well, it sort of kill the whole system. The virus will not be able to replicate in the body of the bacteria. So that is how the defense mechanism in using the CAS system, right? Let me, okay, so there are a few terms that you have to read some more. As I mentioned, this is your assignment, so I'm not going to like tell you everything, but of course I don't know everything. I also still learning. So the term protospacer, the term PAM, okay? So this is tracer RNA, guide RNA. This is something that you have to search for. So let's look at the video, yeah? I think the one that is given by the Professor Mehmet in the assignment. Okay, let me share the video. I think it should be, all right. Okay, so I hope that you can see this video. Okay, let's start.
Now, to begin, we need to talk about the history of CRISPR-Cas9.
Okay,
And it all began the same year I was born, 1987. Researchers in Osaka, Japan, identified in the bacterial genome of E. coli these clustered repeating segments of DNA that tended to be short, but also palindromic, which meant they were read forwards the same way they were read backwards.
Okay.
Now, they also found that these repeating palindromic segments of DNA were interspersed by spaces of DNA that seemed to be quite random. Now, 13 years later, other researchers identified these exact same clustered, repeating palindromic sequences in other bacteria and even archaea. And today we know that they're present in up to 50% of bacteria and 90% of archaea. But back then we had no idea what these repeating segments and even the spaces between did. It took nearly 20 years from that original discovery to identify the fact that it wasn't these repeating segments that were interesting, but the spaces were interesting. Now, what we called this entire stretch of the repeats and the spaces were CRISPR. And CRISPR stands for clustered, regularly interspersed, short palindromic repeats. What these spaces were actually fragments of viral DNA.
Okay, please please take note on what is spacer. It's actually fragment of viral DNA from the virus infecting the bacteria.
Researchers discovered that bacteria had a mechanism to be able to cut fragments of DNA from viruses that were infecting them and incorporate those fragments into their own genome as a memory to remember that invading virus. Effectively, what researchers had discovered was the bacterial version of the adaptive immune system. Let's now take a look at how this CRISPR system works and how we can utilize it for gene therapy. When a virus, like this bacteriophage here, attacks or invades a bacteria, injecting its viral DNA, the bacteria or archaea has evolved a mechanism to be able to identify the viral DNA, chop up certain sequences, and incorporate it into its own genome as a memory of that virus. So an adaptive immune response. How does this work? So firstly, once this viral DNA is injected, the bacteria or archaea has this group of proteins or enzymes called Cas1 and Cas2. So here we've got Cas1, and here we've got Cas2. What they do is they recognize a particular fragment of this viral DNA and they chop it. So it's going to cut it there and it's going to cut it there.
Alright.
What it cuts, this segment here, we call a protospacer.
Okay, remember
We
that,
call
yeah?
a proto.
Because that's one of the quiz. Sorry, that's one of the question. The term spacer, protospacer. Okay.
Spacer. Now, your question might be, where does it chop? Does it just chop randomly or at specific areas? It does chop at specific areas. It will identify a sequence or motif of DNA on this virus that we call a protospacer adjacent motif, also known as a PAM. And once it identifies this PAM region, it will cut a few base pairs upstream of it. So it's now created or cut this protospacer, and it takes this protospacer and incorporates it into the bacterial genome. Once it's incorporated into the bacterial genome, it is now called a spacer. So here we have a spacer.
Okay, so now you have a protospacer, you have PAM, and then once it is incorporated, it is called spacer. Yeah.
From a virus. We've also got another spacer here, which is from another virus. Now, the areas in between the spaces, these are going to be the clustered palindromic repeats. That when a spacer is placed in, a clustered palindromic repeat is placed afterwards. So I'm going to call these repeats. Repeat and repeat. What we call this entire collection of DNA where we've got various repeats and we've got various spaces, we call this a CRISPR array. So this whole thing here is a CRISPR array. And so if we break that term CRISPR up, right, clustered, regularly interspaced short palindromic repeats. Clustered because it's in a particular grouping or area of DNA, regularly interspaced, repeat, repeat, repeat with these spaces in between. Short because they're only like 20 to 30 base pairs long, each of these things. Palindromic because the repeats read the same way forwards as they do backwards. So there we go. This is CRISPR. This is what we're referring to. And here I've only got two spaces, which means this one has actually come from this virus. But this one will have come from another virus. And as I said earlier, some bacteria and archaea can have hundreds of these CRISPR, hundreds of these spaces, so very long CRISPR arrays. So it can store memories of a different virus, yeah? Right? Question is now, what happens? Well, this is double-stranded DNA. Bacteria have circular double-stranded DNA. And what we need to do is we need an RNA polymerase to come into action. So an RNA polymerase, what do RNA polymerases do? They turn this DNA sequence, transcribe it into RNA. Now, in humans, we would usually transcribe this into what we call messenger RNA, which is a message to be able to transcribe amino acids and therefore proteins. But okay, in human, we have DNA, and DNA is transcribed into RNA, messenger RNA. Before that messenger RNA is being used to produce protein, antibodies is an example of protein. So this is in human, yeah, but this is in bacteria. That's not what's happening here. So this is not called messenger RNA. This is actually called pre-CRISPR RNA. So now we've got pre-CRISPR RNA. And as you can see, it is a stretch of repeats and spaces. Wonderful. That's what the RNA polymerase has done. Now, another RNA polymerase will go to another discrete area of the bacterial genome and actually transcribe other types of RNA. These RNAs that it actually creates or transcribes, parts of them match the repeats. Sorry, these parts here. So parts of it will match the repeats. So if we were to draw it up, what we're going to have are these different sequences of RNA where parts of them match perfectly with the repeats and other parts don't. Like that. And so we can put the binding areas in like that, and then other parts won't bind, but then other parts will bind, and then other parts won't. Now, what these strands of RNA are called, they're called tracer RNA.
Tracer
These are
RNA.
called tracer, T-R-A-C-R, tracer RNA. So what we've now transcribed is two RNA strands where we've got a pre-CRISPR RNA and then a tracer RNA that's bound to a repeat, but we've got multiple tracer RNAs. This is where we need to bring in an RNase. So it's an enzyme that breaks up RNA. So what the RNase does is it comes along. So this is the RNase. It comes along and it will chop the spacer and it will chop part of the repeat like that, and it will do it at multiple areas. Now let's just do this one here, right? So we're using this particular spacer, so we've got the entire space.
So I'm not going to finish this with you. You have to watch it and then solve the tutorial by yourself. Okay, so you also need to know about guide RNA.
We can call it guide RNA.
Okay.
So we can actually call these two guide RNA together once it's in the Cas9, gRNA. Okay. Now, in actual fact,
all right, so let's stop share and look at the questions. So I think the first thing that you need to look at is your first assignment called CRISPR assignment. Okay. So this is the link of the YouTube that I have shown just now. Okay. So this is basic. You start with basic structure of DNA and RNA, understanding nucleotide, structure of ribose, deoxyribose. Okay. I think some has been discussed during our class before. ATCG, okay? What is palindromic repeats? You can just Google the definition, try to understand, okay? I think just now has been mentioned what is palindromic, okay? You has the same reading, forward and reverse, okay? And try to answer it. What is the complementary strand of the following sequence? So you just try to solve this if it is DNA and RNA. So since we have issues with like AI software, that's why it has to be written in handwriting. Okay, that's what we have to do now. And as usual, you can convert into a PDF version. I think you don't have issues with this. This is quite easy. And yeah, some have even some answers to this. Okay. I think that it is already some of the guide of the answers are given in the questions. So this is actually by Prof. Mehmet. Both of the assignment is by Prof. Mehmet. Okay. So let's look at the second one, which is a more difficult one. Okay, explain CRISPR. Okay, bacteria defense mechanism. Okay, you explain what is basically CRISPR, the whole idea of the idea of CRISPR and how the bacteria defense mechanism is actually, it can be together. When you explain CRISPR, you can also explain bacteria defense mechanism, but you need to study a bit on how it is being used in biosensors. You probably can give one or two examples. Yeah, the application of CRISPR in biosensors. Okay, Next question is all about terms. So it's not that difficult. You find the meaning of protospacer. Just now you have watched the video. You have it has been explained what is spacer, protospacer, and there is PAM, protospacer, adjacent motif. Okay, but there are also additional, like you have to search what is Cas9, guide RNA. I think it's in the video. Okay, right. There are a few extra things like knockout, knock in, off target. Okay. This is probably a bit difficult, which the challenge one is for you to explain the difference between these three systems, because Cas, the enzyme, and the complex system that edit the gene, that cut the gene, there are three systems, Cas9, Cas12, and Cas13. So you have to explain a bit what's the difference between these three systems. Okay. Right, questions?
Prof, how about the question on write the abstract of two papers?
I have removed that, but I think it's
Okay,
that you just have to write the abstract, so it's just not. Okay, I removed that.
Okay, all right. So the last question would be the explain cis and trans activity, right? That one. Wait. We need to discuss the figure. The figure below this one. We need to discuss it, right, Prof?
The figure is actually the Cas system, so you're actually
Okay.
discussing the difference between these three. So I'm just trying to tell you that try your best, okay? So this is an assignment which contribute 35%. Okay, so your first assignment will supposed to be submitted by this end of June, right? That's what we agreed. So for this assignment, because our exams will be on 10 of July, right? I hope that you know all the dates already. Yeah?
Yes, Prof.
Okay, so ours will be 10 of July, and common practice is I have to submit all the marks within 2 weeks after the final exam, including all the assignment marks and all that. So I'm giving you, if it is 10 of July, so I expect you have to submit this tutorial by 15th of July. That's the end. Means that 5 days, because I have to mark and another 5 days to complete the submission of the marks. Okay?
Yes, that works, Prof. Thank you so much.
Okay. So since we don't have any, if you have question, please ask. I don't have any more. So next week what we're gonna do, I'm not going to have any class because I don't have anything more to discuss. What I will give is just like a few questions for you to just practice by yourself, not to be submit, to question what you have learned from the beginning, like what is receptor, what is transducer. You just answer the questions next week by yourself as a review, as a revision. And you know that the final exam question is open source, yeah, not just open book, open source. Okay. Right. If there is no questions, thank you so much. I think Ahmed would like to discuss something with me.
Yes, Dr. Ahmed.
Okay. So I hope this session helps everybody to answer the tutorial, the assignment. Okay. All right. Thank you.
Thank you, Prof.
Thank you, Prof.
Thank you, Prof.